Hello everyone, welcome to the NN Classroom. Today I'd like to share with you a multiple choice extracted from the Specimen paper 2016, question number 7. A really typical multiple choice asking concepts from a higher level area. Topic 7, nucleic acid. At the last part of the nucleic acid, about the protein structure in terms of 4 levels, right? Let's see. The diagram shows the molecular structure of a human hemoglobin found in the protein data bank. So as you can see, a 3-dimensional picture showing part of the alpha helixes and maybe some beta platelet sheets plus some random coils, right? What characteristic of a hemoglobin allows it to be considered as a protein with a keyword here, quaternary structure? Quaternary structure will be the keyword in this question. Simply applied in the question asking for about the definition behind quaternary structure. For here, students are required to know the details about the protein organization. So just remind you that we first classify protein, or we can call polypeptides, in terms of 4 different levels. The first one, we call it primary structure, and then secondary, tertiary, and finally quaternary structure. Each of the level has a distinct feature or description or definition behind each level. The first one, at the primary structure. Primary structure is actually describing the amino acid sequence. So just remind you that if they're asking for primary structure depends on the sequence of the amino acid sequence. So that means how is the spelling of the residue across the entire strand of the polypeptides. So that is what we call the primary structure. Obviously, it's specific only in terms of the sequence, but not yet the structure in a 3-dimensional picture, right? So secondly, is what we call secondary structure. So secondary structure concern about once I'm finishing the primary structure, I can do a process called folding. Folding is a process where the protein will get specific in terms of the structure. After the fold, the what we call structural domain. D-O-M-A-I-N. Structural domain describing part of the sequence can fold in this way and part of the sequence can fold in that way, usually containing alpha helixes or beta platelet sheets. So those are concerning what we call secondary structure. Finally, tertiary. Tertiary is concerning a very clear, straightforward description. That is to say, a specific 3-dimensional conformation. Anything that has folded to a maturation phase start with tertiary structure, which has a specific 3D shape, normally concerning only one polypeptide. So that's why here of one polypeptide. So you can describe this as a globin with a specific 3D shape, right? So that's why most of the enzymes normally achieve the tertiary structure and would be described as mature or functional as a performance like a catalyst, right? But sometimes protein attaining tertiary structure may not be satisfied, like the case of hemoglobin. So normally some of the proteins will attain even further in terms of the levels of organization. So that is what we call quaternary structure. In a quaternary structure, the focus is that more than one of the polypeptide that already attained with the 3-dimensional structure or tertiary structure. That means they're already specific, but we need more than one of that globin plus some non-protein components, right? So in the case of hemoglobin, a really common example, that's the existence of the heme group, which is what we call a non-protein component. Sometimes we can use to describe them as like some cofactors that cooperating with the polypeptide strands in order to make the whole structure super complicated and really specific in terms of performing specific jobs, like the case of hemoglobin, transporting oxygen. So that's why in terms of the description, the real description for quaternary structure is that we need the protein to be more than one of the polypeptides. So that's why you can see alpha and beta subunits, right? So that's why those are describing now I'm having more than one of the specific globin plus some of the non-protein components. So that will be our heme group. So that's why for this perspective, if you understand the definition behind the 4 degrees of organization, then you can choose the right answer, right? To be C, right? It consists of polypeptide subunits, more than one, plus some non-protein components. So that's why answer obviously B.